Awesome. We're here today at AACR 2025, and we're joined by Tyler Burns. So welcome to Teiko's YouTube channel, and thanks for having us.
It's good to be here.
Yeah, awesome. So real quick, we're just hoping to understand a little bit about you and like how you got into this field, and maybe just tell us a little bit about yourself.
Sure. I mean, if we roll the clock back to 2012, that's when I joined the lab of Gary Nolan. And this was around the time that CyTOF was really coming up. So there was a paper that came out by Sean Bindel and Aaron Simons in 2011, and I joined the Nolan Lab just as, that was the talk of the town, essentially. CyTOF Leipzig that year had Gary Nolan as the keynote speaker. And I would say like a really important thing in terms of my background is I got to see the paradigm shift from, 8-color flow to 30 to 45 markers in a single panel. And that required an entirely new way of thinking about the data. So prior to this, you would be doing manual gating of various cell subsets and then do your statistics on that. But the problem was, if you're dealing with 45 parameters, you have basically 45 choose 2 biaxial plots. And I think that's something like 990, very large number. And so we needed a new way to think about how we would do this. And so part of this is, obviously clustering and that kind of thing, unsupervised learning really solved a lot of problems. But the interesting thing that I think that I don't hear enough people talking about is it's not just the convenience of, SPADE and t-SNE and UMAP. It's like, it's convenient. You get to see the forest and the trees. You hear that term a lot. But I think one thing that the interesting thing to me was at the time, at least in my corner of the world, everyone was talking about, cancer is hard to beat because it is heterogeneous. There's a lot of heterogeneity there, right? Well, CyTOF started to ask the question, what does that mean? What is heterogeneity? And there were tools that would allow you to view your data set as a tree, like SPADE, or a map, like t-SNE at the time. They called it VISNE. But the point is, you could see what normal would look like. And then you could overlay, you could concatenate the normal data with the tumor data. And then you could see regions of phenotypic space, is what we called it, that were specific to the cancer. And what seemed to be the case is that when you looked at different, if you overlaid like a SPADE tree of like normal bone marrow, and then you overlaid a cancer patient, and then you overlaid another cancer patient and another cancer patient, it was all over the place in terms of where the new cells were kind of showing up. But the interesting conclusion that Gary would say in his talks was that cancer was exploring new regions of phenotypic space. So almost thinking of it like how an animal is an intelligent agent that solves problems in physical space. You can almost think of cancer as I got to be careful with my words here, but I'll be a little bit loose here. So cancer being an agent that's solving problems in phenotypic space. And
just for its own survival.
For its own survival. And that was like a new concept that did not really exist prior to CyTOF.
How did people think about it beforehand?
So far as I'm aware, people were thinking in terms of, there are multiple cell subsets here. Obviously, like if you give the cancer patient chemo and radiotherapy and whatever else, and all you need is, one cell out of a billion to have the genetic combinatorics to be resistant, to then divide and then replace the tumor effectively. So, but the question that had not been answered yet is how many cell subsets is a tumor? Right? Or is it all just kind of, is it distinct populations? Or is it all a big smear? We can use these terms now because everyone has seen 1,000 UMAPs. And so I can say, like, is it a bunch of islands? Or is it a big smear? And I can say that and it makes sense, but go back to 2012 and no one knows what a t-SNE map is. This is super new stuff. And so Gary would show these SPADE trees and t-SNE maps, and everybody in the audience was just like this. People don't appreciate, it's obvious, and it's kind of, like how ChatGPT is kind of this retroactively obvious thing. But back then, it's like people forget just how crazy that was.
I mean, UMAP came out in 2019, right? And before that, there was t-SNE and so on and so forth. So, why maybe explain a UMAP in very dumb terms to a simpleton like me.
Sure.
You know, like what is a UMAP? Why would a drug developer ever care about that?
Yeah, that's a good question. t-SNE and UMAP are both nonlinear dimensionality reduction algorithms. And the easiest way to understand this is like, let's say you have your point cloud of cells in high dimensional space. And here's a cell, and here's its nearest neighbors. And there's a way that the nearest neighbors are represented, like a mathematical way. So t-SNE, it's probabilities, and then UMAP, it's using abstract topology.
Yeah.
But either way, it's basically grabbing the nearest neighbors and saying, okay, these are your nearest neighbors, these are your nearest neighbors, these are your nearest neighbors. And then now you have a flat two-dimensional space over here where the cells are prearranged in some like non-random way, like PCA or something like that.
Yeah.
And then there's a loss function. So the nearest neighbors are recorded here, and the nearest neighbors are recorded here.
Yeah.
And then there's basically a loss function that says make the weighted nearest neighbor graph here as similar to the weighted nearest neighbor graph as possible here. And so then it iteratively rearranges the cells. So this difference, you might hear the term cross entropy, you might hear the term Kovac-Leibler divergence, till this difference in effectively the similarity of the distributions is minimized.
Yeah, yeah.
And then that resulting structure has been found to basically have islands where if there's T cells, they form an island here. If there's B cells, they form an island here.
Yeah.
But that's effectively what that is. And so then the question is, okay, why should you care?
Yeah.
Well,
and why would a drug developer care?
Why would a drug developer care? I would say that, I mean, I'll tell you the way I use it.
Yeah.
I would say that is a very convenient way to see what you have early on. So the initial step of just, okay, we ran the data, what's there?
Yeah.
And so it's like if it's PBMCs, then it's like, okay, there's the T cells, there's the B cells. If it's a totally new thing, like let's say you're doing, let's say you have a panel that does like ovarian cancer or something like that. Wendy Fantel did that in the middle 2010s. When nobody else was doing solid stuff. And so then the question that she had was, what's this going to even look like? What's this going to even look like? And she had seen SPADE trees and she had seen t-SNE maps prior to, but she had no idea. Is it just going to be one big thing? Is it going to be like a million things?
Yeah.
Right. And so especially when you're dealing with like super novel stuff.
Yeah.
What do we even see here? Is it a smear? Is it a bunch of like islands? If it's a bunch of islands, maybe we can target them. So that's the kind of questions you can ask. And then the last thought that I'll give you, and this is something that I haven't really heard people explicitly say.
Yeah.
But when you've been in this game long enough, you've seen 1,000 UMAPs of PBMCs.
Yeah, yeah.
There's one in every paper. UMAP or it didn't happen, right? UMAP or it didn't happen.
Yeah.
So check this out.
Yeah.
If you've seen 1,000 UMAPs and another paper comes out that does, CyTOF of PBMCs, and they show the UMAP, and it looks funny. Then you're like, okay, what antibody clone did they use for CD4? Because there's something up with the T cells, and you can already tell something's off.
There's a look, there's a feel.
Just by, yeah. So there's this intuition component of like, if you've seen enough t-SNE maps of blood and bone marrow and whatever else, you can actually use that as like a very quick visual QC gut feeling. Does that look right?
What about reproducibility in UMAPs? Like if you run the same, low dimensional reduction over and over again, like how reproducible would it be?
That's a good question. I just posted it to LinkedIn a couple weeks ago, where I ran t-SNE 100 times with 10,000 cells, and I ran UMAP 100 times with 10,000 cells, and then I turned it into a GIF, and so you can see the whole thing kind of changing around. And what I found was that the islands seem to stay the same, but their position relative to each other does not. So what that means is that if you're dealing with a t-SNE map and there's like an island here and there's an island here, that doesn't necessarily mean that they're as similar as the island that's over here. It could be that they're moving around all the time.
Yeah.
And so this actually caused a bit of a stir on LinkedIn, because there were some people who said, don't run it multiple times because...
Just do it once, it'll be fine.
Well, so to steel man that for a second.
Yeah. If
you run it 10 times,
there's
going to be this step where I like like that one.
Yeah.
You know, and so a lot of statistics, you know, it's like this is you do not want to have a selection bias.
Yeah.
Right. And so then the Hegelian dialectic there is you run it a bunch of times, and then you have a random number generator select the one that you're going to use. Right.
Yeah.
So what I got from that is I think everyone should run their UMAP multiple times.
Yeah.
Just to see, it might be like for bone marrow, for example, it might be that there's a trajectory that stays together every single run.
Yeah.
Or it might look like a trajectory, but it's actually not, and then it splits off and it's moving around.
But so what if it does? Like, why? I mean, if it moves around, isn't that just the nature of the beast?
So it's a question of like how similar are the cells to each other?
Yeah.
And what has been found is that, you know, it optimizes for nearest neighbors. It doesn't necessarily optimize for the T cells are going to be next to the natural killer cells every single time.
Yeah, yeah.
And this is where it gets a little bit complicated because you see UMAP, if you look at the math, when it optimizes, you can think of it as like a neighbor graph where similar cells are being pulled in.
Yeah.
But in that t-SNE just does that. And then UMAP has this other piece to it where distant cells are being pushed away.
Yeah.
So theoretically, UMAP actually preserves global function a little bit.
Yeah.
But if you only read the paper and you only saw the math and you only saw Leland McInnes' SciPy 2018 talk, you would assume that UMAP solved it. Like, no more problems.
Yeah.
What I found from my work is that it solves it a little bit. It does a little bit better in terms of global structure preservation, right? Which would be the question of how similar is one island to another island?
Yeah.
But not perfectly.
Yeah.
And so you still have to, I would say, the generalized advice that comes out of this is you have to check these things yourself. You have to poke and prod yourself. You change the number of nearest neighbors. Run it 10 times. Change whatever hyperparameters you can. Color it by all of the different things.
Yeah.
See if you can find any faults.
Yeah.
And if you do find faults, you at least know what you're working with. You don't want to run into the other camp that I see on Twitter all the time of somebody saying, here's a fault, don't ever use UMAP again.
Yes, I've seen that.
Yeah. I mean, it's the same thing with like, you know, ChatGPT hallucinated,
and
therefore,
the entire thing doesn't work.
Shut down all of the AI stuff. Because it hallucinated
Bomb
once.
the data centers.
Exactly. So like a lot of my work is like, I actually go into the imperfections of these tools, not to say stop using them, but just so you can have a more mature way of using them.
Yeah.
Right.
So gating is still around after decades, and you know, one would think with all the advances in machine learning that gating would be automated by now, but it's not. So
I mean, what's going on there, huh?
Yeah, why not?
I mean, I remember a talk at CyTOF 2012 in Leipzig where there was a guy who basically spent a whole hour saying, everything's computational now, systems biology is taking over. Remember that buzzword?
Yeah.
The bioinformatician's not going to be needed anymore. This was 2012. And, you know, when people saw the first SPADE trees, well, that was the end of manual gating.
But wait, the bioinformatician's not going to be needed anymore?
Yeah, I mean, if you take we don't need manual gators anymore.
Yeah.
If you take that proposition to the extreme, you generalize it all the way.
Yeah.
Oh, everything's going to be automatable with AI.
Well, yeah.
Well, it is a bioinformatician, right? It's going to be a big it's going to be a computer.
Yeah. Everything's computer.
But if we, and I think the way you solve this is by going into the specifics.
Yeah.
Like, you know, you can make these generalized propositions, but they don't really mean anything. So let's go into manual gating.
Yeah.
So manual gating, of course, you have your biaxial and you have a couple of blobs and you draw your gates around the blobs. Flow cytometry people really like to touch their data.
Yes.
Right? Very, very important.
Yeah.
So clustering comes along. We don't need to do manual gating. There's a couple of bullet points there that are really important. The first, and I know this because my wife was doing CyTOF for the clinic in Berlin. And so she was working with a bunch of old German doctors.
Okay.
They want nothing to do with UMAP. They want nothing to do with heat maps. They want nothing to do with the computer. They just want to know the manual gates. They want to see the bar plots with the error bars and then the thing and then the stars, you know.
Yeah.
That's all they want.
Yeah.
That's
and we
all
hear
they
that
want.
a lot of time from translational scientists. It's like, I don't understand the basics. I don't understand what's going on underneath the hood with some of these advanced visualizations, and I understand gating, and I understand all of those, and so I'd rather just use those for right now.
Yeah, exactly. And this was my wife's experience, so she used UMAP as a convenient way to see what she needed to gate down to.
Yeah.
Right? So that would save her time in that she wouldn't have to exhaustively manually gate everything ever.
Yeah.
She would just look at the UMAP and say, okay, do some preliminary statistics, diff site, something like that.
Yeah.
Okay, the natural killer cells matter. Let's gate into the natural killer cells.
Yeah.
And then, so that's how she used it.
Yeah.
And so for her, manual gating still very much matters. And it sounds like, for some of your clients and then some of the clinicians, manual gating is still
crucial.
Gonna be a thing.
It's crucial because manual gating, you can feel it, you can trust it, you understand it, you can actually even see the signals, and you can actually follow the entire chart. And it's like, UMAP, like to rest a multi-million-dollar next stage of your experiment on it, it's still, I think it'll get there, but because there may be tools to bridge the two things.
Well, so at the end of the day, so we're gonna get into a tool that does that. But like at the end of the day, especially if there's a lot on the line, I want to be able to connect the abstract to the concrete.
Exactly. Yeah.
And so if there's a beautiful UMAP that shows me a thing, okay, I want to see the manual gates.
Yep.
Like I will check, and that's fine because the UMAP gave me the convenience of saying I know the populations that should be different, and then I'm going to do the rest of it with the manual gates.
So like complementary as opposed to like pure substitute. Right
Exactly.
now.
Exactly.
Yeah.
Now, John Irish's lab has a tool.
Yeah.
I forgot the name of the tool, but I know it's there, where you do the clustering, and then you algorithmically figure out by some sort of binary thresholding, how do I manually gate down to that cluster?
How would a human have gated to this?
And so that's like a really cool.
That's fascinating.
So I think there should be more of these algorithmic bridges to connect the abstract to the concrete, especially, this is a trend we haven't really talked about, but this is something I'm seeing a lot in my services, that so I was a wet lab guy and then I transitioned to dry lab.
Yeah.
What we are seeing is basically that happening right now in mass, because you have this kind of spin cycle where the PI wrote the grant, underestimated the amount of bioinformatics support he or she needed, and then there's a smart postdoc. Here's $500, take this boot camp. The rest you can use ChatGPT. Maybe we query the bioinformatics core. But I've seen a lot of labs who just need a full-time bioinformatician, that's it.
Yeah.
Right, and so what I'm saying is that there's a lot of people who are being forced into this very technical world. Mm-hmm. And one of the solutions to that problem is to have these tools to make the abstract concrete.
Yes.
If only for them, but I'm as deep as you can get in bioinformatics, I would say, and I still want to bridge the abstract and the concrete. So that's not something that goes away with a certain level of experience. I think it is just something that it just needs to be a foundational bedrock. There's this incentive when you make a new tool to like show it off.
Yeah.
Look at these pretty pictures. It is awesome. Like I have an acronym associated with it. The late 2010s was full of like new algorithms with new acronyms.
Yeah.
But at the end of the day, like, what does it do? How does it help the client?
Yeah.
What problem does it solve?
Yeah.
You don't need to, like, the rest is just kind of glitz and glam.
Yeah.
But at the end of the day, when I look at a new tool, when I really look at it, I open up the hood and I say, oh, phenograph is really just Louvain clustering of a K&N graph. Okay. And then I don't have an obscure word in my working memory anymore.
Yeah.
Because now I know, like, the layer underneath is actually simpler than the abstraction.
Yeah, it's interesting because even in things like batch normalization in cytometry, there are a lot of very sophisticated algorithms, and I think something that worries drug developers is like, is the computer doing something to my samples from run to run?
Yeah.
And so the best is you don't do batch normalization, but if you do batch normalization, then perhaps there are ways to like expose exactly what's happening that that's computationally happening to each sample.
Yeah.
So you can see underneath the hood, just like the example you mentioned in biaxial gating. Yeah.
Well, so the way I do it, and I'm surprised that like I have not seen people do this.
Yeah.
But I had a preprint in 2017 where I effectively had a batch 1 and a batch 2.
Yeah.
And what I did is I concatenated the data together. And then we were just talking about t-SNE and UMAP and like the nearest neighbor stuff. So check this out. You have, so imagine you have your point cloud that contains batch 1 and batch 2. And then you just take each cell and you take their k nearest neighbors, so that you take their 100 nearest neighbors. And then you ask the question, what percent belongs to batch 1? What percent belongs to batch 2? Is it 50-50? Is it 60-40?
Yeah.
You record that for each cell. You color the UMAP by that number.
Yeah.
And what that does is it's basically kind of a batch effect locator. And so it might be that you have a batch effect in like, the CD44 population.
So it could only certain parts of the readout were affected. Oh, that's really interesting. So you can be more surgical about your application about
yeah. And then the, even if it was like even if you find the batch effect everywhere, it's happened. What you can do is you can use that as a proxy for like, okay, I did a z-score, I did quantile normalization, I did algorithm X, I did algorithm Y.
Yeah.
This is a metric to measure how much it corrected the batch effect.
Yeah.
Right, so that's an important thing. But I was just having a conversation with someone yesterday about this topic exactly. So he runs a bioinformatics core at Mayo.
Yeah.
And what he told me was that a lot of the stuff is dataset dependent. So that's one component.
Yeah.
And then the second component that we definitely agreed upon is like, you do the normalization.
Yeah.
And then we go back to, okay, this is the theme we just talked about, bringing the abstract to the concrete.
Yeah.
So what do? Show me the biaxial plots. Show me the biaxial plots. I have a scar, like a business scar, we'll call it. I did a project and I just did like a simple z-score normalization, and then spent a lot of work on it. Showed the biaxial plots to the client, and all the flow cytometry manual gators in the audience. They're like, I don't buy it. Yep. I'm like, why? I'm like, because that marker doesn't do that.
Yeah.
Why is it doing that? And I'm like, well, I did a z-score thing, and it's like, show me the non-normalized by. Okay, those make sense.
Yeah.
Like when you did the normalization, it ruined the biaxial plots.
Yep.
So I don't trust this data. And so that's when I kind of stopped. I had this whole I was trying to basically make a batch, effect normalization algorithm metric thing.
Yeah.
And as soon as I did that step that I should have done in the very beginning of bringing the abstract to the concrete, I said, wow, okay, there's a lot more to it here than like some sort of k nearest neighbor score or anything.
Yeah.
Show me the concrete.
Yeah. Well, Tyler, awesome having you on the channel, and it's great to meet you live and direct, and hopefully we can see you next time in Europe.
All right. Sounds good.
Thank you.
Thank you.